Now we'll start working through the method of translation and the mechanics of it. Some big things to take care of right up front. Translation is actually going to take place on a ribosome. So that ribosome is going to be kind of this bluish green color here. And the ribosome is actually going to be a component that is made up of two different subunits, a large subunit and a small subunit. Those are going to come together and then start the translation process by moving in the 5' to 3' direction. So the same that we've been talking about with DNA replication and transcription. All right, so an overview of transcription is that it really happens in four major stages. The first stage is transfer RNA charging, and this is where the amino acids are going to be floating around inside the cell, and those are going to be charged or attached to the transfer RNAs. We then move into initiation, and in initiation is where all of the components that are necessary for the translation process are assembled at the ribosome. That leads us into elongation, where the amino acids are going to be added to the polypeptide chain. And then finally, termination, and this is where protein synthesis halts and the translation components are then released. So step one is tRNA charging, or where we see a binding of an amino acid to a transfer RNA. And this happens when on the carboxyl group. So if we look at this transfer RNA structure here, we can see that it's made up of ribose material. So we've got our double-stranded ribose material, and on the end of that transfer RNA is going to be where the amino acid is actually going to sit, and it sits based on a bonding that is going to happen. And so keep in mind, the only difference between these amino acids is the R group. So in order for correct amino acid binding to take place on the transfer RNA, we have to make sure that that amine group and the carboxyl group is actually in place. So that transfer RNA is also going to have the anti-coding region down here at the bottom. So that is going to be the opposite code that is going to find its partner on the messenger RNA strand as the ribosome moves along from the 3' to 5', from the 5' to 3' direction. So this is what it looks like chemically. So this transfer RNA, the biggest portion of it is going to be down here in green, and that's what we see over here. So that's where we're going to have our phosphate groups and our adenine going to be bound to our amino acid up here. So the carboxyl group of the amino acid is going to attach to the hydroxyl group of the 2' or the 3' carbon atom of the final nucleotide. And that final nucleotide is going to be on the 3' end of the transfer RNA. So as we look at these transfer RNAs, there is going to be a very specific tRNA for each particular amino acid. So if we look at this figure here, it's kind of like a really bad game of gin rummy. Okay, but there's certain aspects and base pairs of the transfer RNA that are going to be synonymous with a certain structure, and more specifically these hairpin structures. So our anticodon that's going to be at the bottom of this transfer RNA that's actually going to be looking for a very specific sequence on the messenger RNA is going to be an area where we get specificity between the transfer RNAs. So if we have a transfer RNA that codes for serine and another one that actually codes for arginine, the anticodon is going to be very specific for those two transfer RNAs. So another enzyme that is at play here in the TNA charging process is amino acyl transfer RNA synthases. And these synthases, or these different enzymes, really ensure the specificity to take place. Okay, so there's going to be one synthase for each amino acid, and there's 20 different amino acids, therefore there's going to be 20 different synthases. So if we look at this figure again, the positions in blue are all going to be the same in all transfer RNAs, and those areas really can't be used to differentiate one from another. The positions that we see in red are going to be important to the recognition of the transfer RNAs by one synthase. And then finally, again, the positions in yellow are going to be used by more than one synthase. So this is where we get that degenerate code thing going on again. So tRNA charging is really the very first step of this entire process, as we're talking about the translation portion of protein expression. And the overall take-home message of this step is that we're going to get transfer RNA that is going to bind to an amino acid, and this takes energy to do that. So once the amino acid is bound to the transfer RNA, we call that amino acylated transfer RNA, or charged. And so the reason that it's charged is because it takes energy to make that relationship happen. So as we can see on this figure down here, working from left to right, we've got our amino group, so our 4 major components, and our radical R group. That is going to be joined to the transfer RNA over here. But before we get to that point, adenine triphosphate, or ATP, is going to be converted into 2 phosphate groups that are going to come off of that triphosphate structure. So that's where we're losing energy and or losing phosphate. So we're losing negative charges so that this tRNA binding can actually come together. And the product of this is we end up with an adenine monophosphate because there's only one phosphate group left. And after the end of that process, our amino acid is then hooked to our transfer RNA. Once that is complete, that takes us into step 2, which is initiation. So keep in mind, this is going to be very different than RNA DNA replication and transcription, because initiation has always been step 1. But in translation, initiation is really step 2 behind RNA charging. So with the initiation process, this is where all of the different components are going to come together and assemble on the ribosome. So those different components are listed on this slide. So we've got the mRNA strand that came from the transcription process. We've got the small subunit, or the 30S subunit. We've got the large S subunit, which is the 50S subunit. We've got a bunch of initiation factors. Those are labeled as IF3s here, and there's more of them depending on if we're talking about bacterial species or eukaryotic species. We've got our initiator transfer RNA that was just charged in the first step, and then we have guanine triphosphate, which is going to be our energy. So when all of these components come together, they're going to assemble on this ribosome. So this ribosome is going to go through and is going to be hunting for this messenger RNA strand. Then the the smaller subunit, the 30S subunit, is going to attach to the bottom of the messenger RNA first, and then we're going to get the second bigger subunit to come in a little bit later. But in order for the bigger subunit to come into place, the ribosomal cell subunits have to exist in the cell. And these guys are always, always, always moving, because if we think about entire chromosomes that could contain upwards of 10,000 genes just on one chromosome, there's lots of ribosomal material in place already. So these guys are just kind of floating around the cell looking for messenger RNAs to come together with initiation factors. So IF3 in this case, or initiation factor 3, after the subunit, the small subunit kind of finds the messenger RNA, IF3 is going to bind to the small subunit of the ribosome, and that's going to allow the messenger RNA to then bind to the small subunit. After this takes place, but before the large subunit comes into play, we need to make sure that the Shine-Dalgarno consensus sequence on the messenger RNA is complementary to a portion of the small ribosome. And that is what really is going to allow further binding to take place. So the Shine-Dalgarno sequence is going to be upstream of that initiating methionine. So if you keep in mind AUG, the code AUG always codes for methionine, and the Shine-Dalgarno sequence is going to be upstream from that initiating methionine. After initiation factor 3 has attached to the small subunit, and then the messenger RNA has attached to the small subunit, and we found the Shine-Dalgarno sequence, then we can start using guanine triphosphate, or GTP, as our energy to bring in initiation factors 2, initiation factor 1, which are going to help bind to the small subunit and the transfer RNA respectively. And this starts to bring in all these factors that are going to help stabilize this bigger structure that we're building. So it's like building blocks. All these things have to come together. In the meantime, we're also going to get the first transfer RNA that's going to have the anticodon UAC, which is going to be the anticodon for methionine. That's going to come in and assemble against the methionine codon here. So once it assembles, then IF1 is going to come in and bind to the bottom of the little subunit. And the mRNA is now officially positioned correctly. And the initiation factor 1 is going to also associate with the large subunit as well. So once we have all these things in place, then that large subunit is going to come in after IF3, 1, and 2 leave, and we get some energy exchange here, and is going to sit down on top of the transfer RNA and is going to start moving from the 5' to 3' direction. So don't get too confused, because over here we've got listed the small subunit as 30S and the large one is 70S. It goes back and forth between 50 and 70, but the larger ribosome, depending on what species we're talking about, is always going to be much larger than the smaller ribosome. All right, so the initiation complex is now in place, and in the next segment we'll move into elongation and termination.